Welcome you to this session. We have chairpersons with us, Dr. Anil Limai, and I welcome you, sir. He is Associate Professor at the Department of Biosciences and Bioengineering, Indian Institute of Technology, Guwahati, Assam. His areas of interest are hormonal regulation of gene expression, endocrine cancers, and mechanisms of action of bioactive compounds from natural sources. He has a PhD in medical endocrinology from Indian Institute of Science, Bangalore, and he is a member of Indian Association for Cancer Research. We are happy to have you here with us today, sir. Our second chairperson is Professor Vijay Rani Rajpal. She is Professor in the Department of Botany and Raj College, University of Delhi. Her areas of interest are cytogenetics, molecular markers application, population genetic analysis, and phylogenetics. Professor Rajpal has a PhD in botany from Department of Botany, University of Delhi. A postdoctoral research scientist she was at Department of Botany again. She has been a lecturer in botany at Acharya Narendra Dev College, University of Delhi. She was awarded 33rd Dr. Radha Krishnan Memorial National Best Teacher Award in the year 2017 for exemplary contribution in the field of education. She is also a recipient of Bharat Ratna Dr. APJ Abdul Kalam Memorial Grace India Best Teacher Award, which was given to her in October 2018. She was also awarded Grace India Best Researcher Award in 2018. She is a member of New York Academy of Sciences, USA, and has edited 4 research books and authored 4 book chapters of national and international publishers. She has published 20 scientific papers in peer-reviewed international journals. With this brief introduction, I welcome Professor Limai and Professor Rajpal to chair this session and initiate the proceedings. So all the presenters, they get 10 minutes to present. After 8 minutes, they will be reminded that 2 minutes remain, and after which I will again inform that the 10 minutes are over. Following which there will be a short Q&A session.
We welcome you to this session. So it's a young brigade of the upcoming scientists of India. So it's great to see you. We have come a long way since the time of phytochemicals like morphine, plasmine, and others. They found use in various disorders related to humans. And nowadays, as I attended a session in the morning, that we have a whole field of Ayurvedic genomics coming up, which is sponsored by Ayush. So we have a lot of opportunities for drug discovery and alternate system of medicine, especially related to phytochemicals. And you can definitely contribute immensely, and you can carve a niche for yourself. So I welcome you to this session and wish you all the best. This is just one request. We are already delayed by around 45 minutes. So you are requested to stick to the timeline. So try to finish if possible in 8 minutes. But of course, the time allotted to you is 10 minutes. So 8 minutes the buzzer will ring, and 10 minutes sharp you should close. So that is some time for discussion also. We have our co-chair in the form of Dr. Limai. So if you allow some time for discussion, then you have his inputs also coming up, which will be really helpful to you. So all the best to you. So here we begin. I think we can call the first presenter, or if Dr. Limai wants to make some remark.
Anyway, without wasting much time, let me call upon speakers straight away. We have S1c Valsan, and she is going to speak on S1c, a compound isolated from Cyclia pentata.
So, respected dignitaries chairing the session, other research scholars, S1c OP1 alkaloid, which is isolated from cyclia pellitator that induces a mitochondria-mediated apoptosis in cervical cancer cell and HeLa. The work was done under the supervision of Dr. K. V. Radhakrishnan and Dr. Kaustubh Kumar Maiti at Organic Chemistry Section, CSIR NIST, Toronto. Cancer is a group of diseases involving abnormal cell growth with the potential to invade or spread to other parts of the body. According to WHO reports, cancer is the second leading cause of deaths worldwide, accounting for nearly 10 million deaths in 2020 alone. There is a report from American Cancer Society that from 2016 to 2030, the worldwide cancer cases are projected to increase by 24%. That is exactly from a number of 17 million to 21 million. So there is an urgent need to develop more successful therapeutic entities with affordable cost to cure this deadliest disease. So natural product is a better option. Nature has generated an astonishing array of molecules with diverse forms and functions, refined by evolution over billions of years. Natural product is any organic compound that is synthesized by a living organism. The identification or the development of a drug from a natural or plant-based origin is a tedious process which involves the isolation, characterization and the screening of many bioactive molecules. And the identification of a natural product for an in-depth study to develop a natural product-based drug is a need of the hour today. So keeping in this mind, we have selected a plant which is known as Cyclia pellitata, belongs to the family Menisphaceae. The plant is known as Rajapata in Sanskrit and has been a part of many formulations in Ayurveda, especially for the treating ailments of women reproductive system ailments. So from the available pieces of literature, it is clear that the plant possesses some anti-cancer activity, but it is being studied only at an extract level. So we decided to extract the phytomolecules which is responsible or which is imparting the anti-cancer potential, and successfully we have isolated 3 compounds from the rhizome of the plant which are fiandin, cyclianin and cordyalin. And we carried out the preliminary cytotoxicity screening by entity assay, and from the table it is clear that the compound fiandin possesses more potent inhibitory activity against cervical cancer cell HeLa with an IC50 value of 8.1 micromolar. Next, we are interested whether the compound is safer to a normal cell line up to a minimum concentration that can be used in a cervical cancer. So we have selected a cell line MCF10A, which is a non-tumorigenic cell line, and in the cell line we could see that it is safer up to a higher concentration, and its IC50 comes only up to 36. We are not using that higher concentration. So we have selected this plant for our downstream in vitro studies. First, we studied whether our compound can inhibit the clonogenic potential of HeLa cells, and from the result it is very much clear that at a very low concentration, that is 0.5 micromolar and 1 micromolar itself, the compound is able to inhibit the colony formation ability of individual HeLa cancer cell lines, and also it downregulated or reduced the survival fraction at a higher good level. Next, apoptotic studies, we use some microscopic techniques like light assay and APOP staining, and from the result we could see that more number of red to colored cells in the first panel, that is this figure shows the more number of apoptotic cells, and in the APOP staining we could observe purple stained cells in the treatment group showing the membrane compromised cells due to apoptosis. To confirm the process of apoptosis involved in the process, we have studied the Anaxin-5 assay, and from the result we could observe a more population of cells in the late apoptotic cells upon treatment than the control group. Also, we performed the caspase fluorimetric assay to study the involvement of which caspases, and we studied the expression of the executional caspase 3 and other 2 initiated caspase 9 and caspase 8, and from the data we could observe a upregulation of caspase 3 and caspase 9, which clearly indicates the involvement of an intrinsic apoptotic pathway. Since mitochondria is involved, we were interested in our compound, whether our compound can induce some depolymerization. First, we studied the mitochondrial depolymerization by JC1 assay, and from the result we could see the JC1 aggregate to monomer ratio is getting considerably reduced upon treatment with the compound, and also we studied the release of cytochrome C from the mitochondria into cytosol, treatment with the compound fiandin, we could observe a release or downregulation of mitochondrial cytochrome C and an upregulation of the cytosolic cytochrome C, which clearly indicates the release of cytochrome C from mitochondria into the cytosol upon treatment with the compound fiandin. And nuclear condensation and fragmentation are two more important parameters of apoptotic DNA. So we studied the condensation pattern mainly by nuclear staining, and in this image we could see a more brightly fluoresced areas which indicates the condensed DNA. Also the fragmentation pattern was observed by a peculiar DNA fragmentation pattern, which labyrinth pattern, which is a characteristic feature of apoptotic DNA. Also we studied the internalization and DNA fragmentation by surface enhanced Raman spectroscopy, and we could observe the characteristic peaks of fiandin at these areas, that is 1000 cm inverse, 1212 and 1590 nanometer are getting reduced or diminished after an incubation of 4 hours, which may be because of the metabolic degradation inside the cell, and also DNA fragmentation pattern was again confirmed using search modality. Next we were interested the compound whether it can induce the cell cycle arrest, and we could observe the downregulation of major key proteins involved in the regulation of cell cycle like cyclin A2, cyclin B1, CDK2 and CDC25. Also from cell cycle assay we could see a probable arrest at an S phase. Also we could observe an increased population of cells at sub-G1 phase, indicating the apoptotic population of cells. Next we were interested in studying the expression of various proteins involved in apoptotic pathways, and we followed this mechanism like apoptotic membrane array, and also we performed the Western blot analysis, and we could observe mainly the upregulation and downregulation of major proteins involved in the pathways. And from the above result, we proposed a mechanism by which the compound fiandin can induce the cell cycle apoptotic cell death. So, firstly, the compound can induce the activation of major pro-apoptotic proteins, and also it downregulate the expression of major inhibitors of apoptotic proteins like XIAP and CIAP, and which in turn activates the caspases involved in the process, and which will activate the apoptotic stimuli and further cell death. And also the compound can inhibit the survival pathway or the survival and proliferation pathway by inhibiting downregulating the CD40 ligand binding, and also downregulating the expression of IGF2, which in turn downregulates the AKT-PAKT pathway, and which obviously it will downregulate the proliferation. Also the compound is able to upregulate the P21, P27, P53 characteristic proteins involved in the apoptosis. So this is a mechanism we proposed by which our compound can activate the apoptosis in cervical cancer cell, right? And as in conclusion, fiandin inhibitor the proliferation of cervical cancer HeLa with an IC50 value of 8 micromolar. It induced a mitochondria-mediated apoptosis and release of cytochrome C into cytosol. Downregulated the PAKT survival pathway, which was we can see from the expression plot of PAKT. Internalization of fiandin into the cells monitored by cells, and fiandin inhibited the chronogenic potential of HeLa cells at a very low concentration, and so it induced the cell cycle arrest. And this is the first report on the detailed in vitro assessment of apoptosis induced by the compound fiandin. Thank you.
Congratulations, Anisha. You have stuck to time. Very good. So we have maybe 2 questions from the audience. Everybody, want me to ask any questions? Everything is understood. Just for the sake of my understanding, I will ask you 2 quick questions.
Yeah.
So in which solvent do you dissolve your compound?
Actually, we performed it in DMSO. DMSO like concentration, and we diluted it in water. Distilled water.
Okay, so you must be having a control, right?
Yeah.
A control dish which you must have a vehicle to do. Right? No?
No, sir. Actually, we use to control as the unfiltered cells of the compound.
So as much as I understand, you are not using a vehicle to do.
Yes.
Because your vehicle itself may be doing something to this. Right?
Yeah, that's why we have minimally reduced the volume of DMSO, and we have diluted all our concentration.
So what is the final concentration of DMSO that we see?
It's 0.1
Good. Last question. You measured IC50 value, right?
Yes, sir.
How do you measure IC50 value?
Sir, we will perform the cytotoxicity assay in different concentration. Like in my case, I have selected 100 micromolar, 50 micromolar, 25 micromolar, 10 and 50. And we will plot the graph, and over 50 comes, we will plot it in.
So that inhibition at 50.
50.
Just by visual inspection, or you use some curve fitting algorithm to find out what is the IC50?
Not algorithm, sir. We calculated using the graph, like at which concentration 50% of cells may get that.
Are there any known inhibitors known for phenol?
Inhibitors. No, I'm actually